Most people imagine sperm as tiny swimmers racing toward an egg. But almost no one imagines where they come from, the hidden place where every sperm begins its life. Deep inside the testes lies a world you can't see with the naked eye. A labyrinth of microscopic tunnels curled and folded like threads of silver. If you stretch them out end to end, they could span hundreds of meters, all packed into a space small enough to fit in the palm of your hand. These are the seminiferous tubules, the birthplace of sperm, the biological factories where the earliest spark of male fertility begins. At first glance, they look empty, silent, still. But zoom closer and you'll see something astonishing. Waves of developing cells arranged like rings of a growing forest. Young cells at the edges, older cells drifting inward, each moving slowly toward the center of the tubule where their journey will truly begin. Inside these coiled chambers, a remarkable transformation takes place. A simple stem cell divides, reshapes, folds its DNA, and evolves into a fully formed sperm cell. A process so complex it takes nearly 70 days to complete. And guiding all of this are Sertoli cells, towering, nurturing structures that cradle developing sperm like microscopic guardians, providing nutrients, structure, and protection. These tubules are not passive pipes. They are living ecosystems, the starting point of a journey that will eventually travel miles, face 5 barriers, and end in a chance at creation. Today, we're going to step inside this twisting labyrinth to watch the very first steps of sperm formation, from the stem cells that begin it to the delicate choreography that shapes their destiny. Let's enter the tubules. To understand the seminiferous tubules, we need to shift our perspective, from the idea of a testicle as a simple organ to the truth that it is one of the most sophisticated manufacturing systems in the human body. Inside each testis lies 200 to 300 lobules, and inside each lobule sits a tightly coiled tube, the seminiferous tubule. Not one, not two, but nearly 1,000 tubules in total. Each one a production line where sperm are created step by step. Under a microscope, the tubule looks like a hollow cylinder, but its walls are alive with activity. The outer edge contains spermatogonia, the stem cells, the raw ingredients of sperm. These are the earliest cells, the ones that will divide and differentiate again and again until they become something entirely new. As you move inward from the wall toward the center, you're not just looking at cells, you're looking at time, each layer representing a different stage of development. The tubule becomes a living timeline. Immature cells on the outside, fully formed sperm near the lumen, ready to be released. And orchestrating this entire process are the legendary Sertoli cells, tall, branching structures that stretch from the base to the lumen like ancient trees in a microscopic forest. They nurture, they regulate, they form tight junctions to create the blood-testis barrier, a protective shield that keeps developing sperm safe from the immune system. Around the outside of the tubules lie Leydig cells, the factories that produce testosterone, the hormone that fuels the entire process of spermatogenesis. Without testosterone, the tubules fall silent. Every part of this architecture is intentional. Every layer has a purpose. Every cell is positioned with precision. But all of this, the structure, the layout, the guardians and hormones, is only the stage. because the true story unfolds when the earliest germ cells begin to divide, transform, and embark on the long journey from simple stem cell to a specialized cell engineered for one mission. And that transformation begins now. Now we travel into the very heart of the seminiferous tubules, into the unfolding choreography known as spermatogenesis, a process so intricate that it turns simple stem cells into one of the most specialized cells in the human body. Everything begins with spermatogonia, the foundational stem cells that sit along the outer edge of the tubule. They divide slowly, deliberately, some maintaining the stem cell pool, others stepping forward into the long developmental journey ahead. As soon as a spermatogonium commits, it becomes a primary spermatocyte, a cell preparing for the most elegant division nature ever invented: meiosis. Here, the DNA inside doubles, not to make the cell larger, but to set up a kind of genetic reshuffling. During meiosis I, chromosomes pair up and exchange segments. A beautiful microscopic dance that creates diversity in every future offspring. By the end of this division, the primary spermatocyte splits into two secondary spermatocytes, each with half the chromosome pairs. But the process isn't finished. Meiosis II follows quickly. The secondary spermatocytes divide again without replicating their DNA, producing 4 spermatids, small, round haploid cells carrying only half the genetic blueprint. And this is where the true transformation begins. Spermatids must now undergo spermiogenesis, a dramatic reshaping that sculpts them into sperm. Their DNA condenses into a tight, streamlined nucleus. A tail forms, engineered for propulsion. Mitochondria gather around the midpiece to build an energy core. The acrosome develops, becoming a molecular piercing tool for future fertilization. Through every stage, Sertoli cells act like architects and guardians, providing nutrients, clearing debris, guiding the movement of each developing cell toward the center of the tubule. By the time the process is complete, the once round spermatid has become a sleek, polarized sperm cell. A remarkable structure built over nearly 70 days. But even now, these newborn sperm are not ready for action. They cannot swim, they cannot fertilize, they cannot survive alone. To gain those abilities, they must leave the tubules and begin the next chapter of their journey. And that transition, from formation to activation, is where the climax unfolds. This is the turning point, the moment when the seminiferous tubules finish their work and the developing sperm step across the threshold from creation to preparation. Inside the tubules, sperm were shaped, sculpted, and refined. But they were not yet alive in the way we imagine them. They couldn't swim, they couldn't sense, they couldn't fertilize. They were passengers, carried, protected, nurtured. Now, everything changes. Once spermiogenesis is complete, Sertoli cells release the young sperm into the lumen of the tubule. This release is called spermiation, and it is the closest thing in biology to a graduation ceremony. The newborn sperm drift into the current created by fluid secreted inside the tubule. Not by their own power, they are still motionless, but by a gentle flow engineered to move them forward. The seminiferous tubules converge into straight ducts, then merge into the rete testis, a network of microscopic channels that acts like a biological switching station. From there, the sperm travel into the efferent ducts, which use tiny hair-like cilia to pull them upward and deliver them into the next great chamber of their development, the epididymis. This is where the climax truly happens. Because the epididymis is not just storage, it is transformation. A three-part corridor, head, body, tail, where sperm gain motility, structural integrity, and the molecular tools required for fertilization. Here, proteins fold into the right shape. Membranes stabilize. Tail movement activates. Surface receptors finish forming. The sperm that entered this tube is not the sperm that leaves it. What began as a shapeless germ cell is now becoming a functional, purposeful traveller. One capable of sensing chemical gradients, swimming with precision, and surviving harsh environments it has never encountered before. The seminiferous tubules created the blueprint. The epididymis awakens the capacity. And it's in that awakening that the deeper meaning of this entire journey begins to emerge. When you step back from the microscopic choreography of the seminiferous tubules, a quiet revelation begins to unfold. Sperm don't begin their life as the streamlined swimmers we imagine. They begin as potential, raw, unshaped, dependent on the environment around them. Every transformation inside those coiled tubules is slow, intentional, guided by cells and structures that exist solely to shape them. The Sertoli cells holding them like scaffolding. The Leydig cells fueling the environment with testosterone. The tubules themselves arranging each generation of germ cells in perfect concentric layers. Nothing about this environment is accidental. Every detail is purpose. And that purpose teaches us something profound about biology. Life rarely starts in its final form. It begins unprepared, unrefined, a blueprint waiting for structure and guidance. Inside the tubules, a spermatogonium doesn't yet resemble the future traveller it will become. It divides, reshapes, condenses its DNA, builds a tail, forms an acrosome, and learns, slowly, how to take on its mission. 70 days of silent transformation before a single moment of action is even possible. There is humility in that. The idea that even the smallest unit of future life must undergo patience, sculpting and preparation long before it can take its first step or its first swim. And when the young sperm finally leaves the seminiferous tubules, it leaves not as a finished being, but as a work in progress, entering the epididymis for its final refinement. The tubules do not create perfection, they create beginnings. And in that quiet truth lies something universal. Every journey, whether cellular, personal, or human, starts in a place of guidance, protection, and gradual becoming. A reminder that creation is not a moment, it is a process.